And next we have Christina Bennett, who's going to talk about assessing the outcome of introducing a digital image quality control review into the publication process for research articles in physiology journals.
Right, I'd like to first thank the organizers for this opportunity to share with you our analysis of our Society for initiating a quality control check for images prior to publication. Our study sought to assess whether the QC check was effective in identifying and correcting images with modifications prior to publication, and whether returning authors who received a QC check once were repeat offenders. The American Physiological Society publishes 15 journals. What I'm going to speak on today will focus on the 7 AJP titles. These are primary research journals, and they combined publish about 15 to 2,000 articles each year, or 1,500 to 2,000 articles each year. And many of our authors utilize photographs to represent their data. These photographs often are images of cells, tissues, organs. They often show expression of DNA, RNA, and protein in the forms of blots and gels. And I would say several years ago, our art department, who we have in-house, started to notice that there were image modifications within the figures that they couldn't tell whether they were appropriate or inappropriate. Around that time, the Journal of Cell Biology, at that time led by Mike Rossner, provided some guidance for journals to look for images and to see what types of modifications were within them. We updated our information for authors and gave guidance to our authors that we would look for evidence of splicing, or that is rearrangement of a particular photograph, extreme contrast adjustment, which is either altering the brightness or contrast to an extreme. We'd look for selective editing, which is altering a portion of an image that isn't done equally across the entire photograph. And we'd look for duplication, either using the same image to represent different experimental results within one figure or somewhere within the rest of the manuscript. As I mentioned, we do have an in-house art staff, and so they utilize the forensics tools that were developed by the Office of Research Integrity at the Department of Health and Human Services to get a forensic, kind of like a fingerprint of photographs to see how authors prepare their data. Because this was an art staff role, they began to do these checks as they were preparing the figures for final publication. However, when concerns were identified and corrections could be made, it would result in us having to post some type of core agenda, because by the time the figures are being prepared and analyzed, the article is already posted online as an early view publication. This was frustrating to both the authors, who were making somewhat possibly minor corrections, and to staff, because they were doing an extra step within the publications process. So we moved the QC check to after acceptance. And so now our authors receive the acceptance letter, and the manuscript file moves into a new little box within the manuscript submission system, which allows our art staff to do the assessment for image modifications prior to the article being posted online or final published. And this allows us to get revisions to non-fraudulent modifications prior to publication. So the data I'm going to talk about today spans over from 2009 to 2016, because it took us several years to initiate this QC workflow. And as you can see, in 2009, there was no formal workflow. And this was about the time when the art staff was noticing modifications, and our leaders at the APS, including our director of publications and the publications committee chair, realized that we needed to take a closer look at the images prior to publication, and we needed to find a way to do this quickly so that we would not delay the publication process. And one of the things that they added was my role as an ethics manager to handle these types of queries. I do have a background in research, and I could look at the science, whereas our art staff could look at the modifications. And if we thought we needed to seek corrections, there would be somebody in-house to answer the scientific issues related to the concerns. We also saw this as an education tool. Our template letters that we use go out to the corresponding author. We notify them of the concerns that we've identified. We state what our information for authors guidelines note about that possible error, and then we ask for corrections along with the original capture. If they provide a correction and if they provide the original capture and it shows that nothing has scientifically changed and it was just a minor modification, then we allow the correction to go through. So it took us several years to onboard all 7 journals, but by 2013, all of the journals were within the workflow. We had a banner year in 2011, so out of approximately 2,000 articles that were published that year, 8% of them received a QC letter from us asking for corrections. However, since then, year on year, we've had a reduction in the number of QC cases for image corrections. One of the questions we wanted to look at was how we had fared with the request for core agenda versus giving the authors the opportunity to revise prior to publication. And as you can imagine, now that the workflow was, once it became all in place, our number of core agenda went down significantly. So much so that in 2015 and 2016, we did not miss any articles up until so that they did not need a core agenda. All of them were, if revisions were appropriate, they were invited to revise their work prior to publication. That's not to say that we allow revisions for all concerns that are identified by this QC check. Instead, we do identify concerns every year that are serious enough that we rescind the acceptance and issue an ethical rejection. These include findings that the images were either falsified or fabricated. And in those cases, those are the red bars. We've retracted articles at the AIPS version for fabrication and falsification. And we've also rescinded acceptance and issued ethical rejections for articles where they could not find the original captures, and we weren't convinced that something untoward didn't happen, or the data has changed in some way such that it requires them to repeat the experiment and go through review again. Another question we wanted to look at was whether our types of image issues that we identified had changed over time. So as I mentioned, we looked for evidence of splicing, extreme contrast adjustment, duplication, selective editing, and compound, meaning a combination of one or more of these issues. In 2009, our art staff identified many issues of splicing, and that was the major thing that they were looking for. However, as time went on, they began to get more savvy with identifying that extreme contrast adjustment was another way to mask modifications. And so now it's quite common for us to identify errors related to splicing and also extreme contrast adjustment. And we always have our handful of duplication and selective editing, but those vary year by year. And then another question, because we made this, we wanted this to be an education tool for our authors and wanted our template letters that went out to authors to be a way to educate them on good presentation practices. We wanted to see if our authors who received these queries and submitted to us again were repeat offenders. So we took a snapshot from 2013 to 2015. This is when all of our journals were now in the QC check process, and looked at whether those authors had submitted again. So out of those 326 authors who received one QC query, 58% submitted another article by the end of 2016. and only 8 out of 190 of those were involved in a subsequent QC query. And we like to think, although this can't be fully verified, that our authors are learning. I think by being part of a scientific society and having a member and good author base, we do have an opportunity to educate our authors and to encourage them to do better practices. And the other caveat I will say is that I did not look to see whether their second paper contained the same types of images as were in the original paper. So in conclusion, implementing this QC check to review image modifications in accepted articles has resulted in the reduction in the number of post-publication corrections. So it makes me sad that we don't often hit retraction watch as much as we could. And the publication of more images as they were originally captured. We do detect serious errors and fabricated data now prior to publication. And we do support our authors by educating them on the appropriate digital image presentation practices. And what's encouraging is that we are seeing a decrease in the number of QC queries year on year, which suggests that our returning authors do adhere to these guidelines, and we look forward to tracking this data further.
Thank you. Just see who gets to the mic first. Okay, go ahead, Ivan.
Thanks, Ivan Oransky, who was apparently born after 1994. But, I'm delighted. My internist will be even more delighted. Really interesting presentation. Of course, you wouldn't be surprised to hear me say that. And I don't know that there's a way to actually look for this. But I'll ask, and you can tell me there wasn't. Do you know what happened to the author? You said 58%. Obviously, a lot of authors came back even after the QC check and what have you. Do you know what happened? Overall, did everybody who had a QC check come back? I'm not articulating the question well, but did fewer of them come back than you would have expected based on sort of typical patterns of submission to your journals?
Yeah, so I didn't look at the trend of non-QC check authors versus QC check. But within a 4-year span, having 60% submit a second paper seems pretty reasonable for biomedical research.
Thank you.
Chris Papazian, Kansas City. So for those manuscripts that you ultimately decided that there was some serious problem with the image due to fabrication or inappropriate manipulation, aside from just rejecting the manuscript, is there any other action that you took with those authors?
Yeah, so we do present their cases to our publications committee to determine whether authors shouldn't be uninvited from submitting work to our journals for a period of time. We also informed the institution many times about these issues.
Becca White, Journal Neurosurgery. We don't deal with these issues at the journal where I am now, but before I was at a stem cells journal, and we saw this quite a bit. And we're a very small journal with very small staff, and so our department was one person. Mm-hmm. And we certainly couldn't afford to have a full-time staff member who was in your role. Mm-hmm. So what would your suggestion be to smaller journals? Because this is a huge issue, and you do need someone who is trained in the science and in the art to be able to be doing these checks.
Yeah, so one of our concerns was putting burden on the peer reviewers, who are also scientists, to do any of these types of checks. But there is something to be said about having a scientist know what the modification is and whether it's worth pursuing or not. So one of the things that we initially told our editors and editorial boards was to use the PDF and zoom in. Maximize the photograph to 400%. So when you're looking at the figures, if things are duplicated, if things have hard lines through them, they should be visible to the naked eye. The forensics tools are fantastic, but we don't want them to become investigators during peer review. They need to focus on the science, but ask them to zoom in. I mean, it actually sometimes frustrates me that something went through peer review and got accepted and no one saw some of the modifications.
I'm Barbara Goldman from American Society for Microbiology Journals. Christina, thank you. That was a very interesting presentation, very valuable. At ASN, we do very much the same thing and have had a similar experience. The challenge that we have now is that the ORI forensic tools are terrific for detecting pixel differences from figure to figure, image to image. And the challenge has been in blots and gels, duplication of bands, because they all look the same pretty much. But there are some amazing individuals with visual acuity that is astonishing, who are able to scan figures and recognize duplicated blots within hundreds of different experiments, including from one paper to another. And I'm just wondering if you have had any experience in tools that help you identify the duplications, which we really have not been able to find. Yeah,
so we do rely on the forensics tools. So I do some screening at times, but Eric Pessinelli, who's our editorial art manager, has Eric eyes, as I call them, and he sees duplications like nobody can. So it is something about being trained and looking at images. The more and more you look, the more and more you think, oh my gosh, that looks really similar, and it looks even more similar, and you just get the eye. So as practice, the more practice you do, the more better you'll be able to see the slight changes. And, it's biology, so every blot should not look the same. And that's when you do, it's when you use the forensic tools just to make a slight modification, you start to see the similarities come out. But it's not easy. Yeah.
Well, hello, Jennifer Byrne from Sydney in Australia. You're probably familiar with the study that was done by Elizabeth Bick that I think was published in 2016 in MBio, which looked at a very large number of publications and I think identified differences in geographic distribution of publications that included problematic images. I was wondering whether you saw any evidence of that in your study.
We don't usually assess it by image modification, but overall we tend to have a 50-50 US versus non-US breakdown of ethics cases. So I don't think it's one country or, one area that's the biggest offender for these types of issues.
Hi, Juno Shiro, Mayo Clinic. I was wondering, you mentioned duplication, and I think from what you were saying, you're saying within a single paper, but is there an easier way to look at, for example, comparing, what's in the current paper versus the author's body of work that came before? Is there some non-manual way to do that?
If you can rely on PubPeer, which seems to be much better at, having sleuths look at a corpus of work. But no, I think our peer reviewers identify duplication better when it's throughout a body of work than we do, because we aren't going back to doing a whole, and we focus on the article that we have. Right. And I wish we could. I wish we had a database that we could put all of our images in and find them again if they're being reused.
And is your in-house process, is that automated, or is it still people looking at individual images running individual tools?
No, it's still manual. We use tools, but that's why it took us several years to get this QC check incorporated, because we did not want to slow down our publication process. And it takes training of, whoever's doing that to get good at it and quick enough to go through these images within 10 minutes versus an hour, which is when we started. It took forever.
Okay, thank you.
Yeah.
Hello, Michael Todd, Innsbruck, Austria. Forgive me if you answered this during your talk, but I'm coming at this from the medical writer perspective. And I do edit images with papers, never unethically, of course, but I'd rather it be clear and not be asked to address the ethical issue. So if I'm submitting a paper, should I always tell the journal? What would you like to see from the medical writers? Should we tell you we edited an image?
Yeah, if authors do a non-standard modification, it would be good to use some legends to say that I'm presenting 6 out of the 12 lanes for this particular image because these are the ones that are pertinent for the study, and I'm indicating this with a space. Or I've my photograph has extreme contrast to highlight this one aspect. Tell us what you're doing. Otherwise, it may look like you're trying to sway the data in a way that you don't really intend to do so.
So I just put it in the legend that it was edited.
Yeah,
all right.
That's what we encourage, yeah.
Okay, if there are no more questions from the floor or online, I do have one more question for you, sorry. Just related to that last question, you mentioned that over the years the authors have become better at knowing what they can and can't do. Do your instructions for authors provide detailed guidance for what can be done and what isn't acceptable?
I will say that they're more of can'ts, you know, that they can't splice without declaring it, they can't over-adjust, but we do encourage them to declare more in the legends if they do need to do some form of modification.
Okay. Thank you.